pleasure today to okay. It's a pleasure to welcome today Mark Lever from University of Texas at Austin. Mark, he just get the associate professor position in September at Austin, and previously he was at ETH Zurich, where he was an assistant professor. So Mark is an interdisciplinary biochemist and microbial ecologist, and he's interested in all aspects of time scale of the carbon cycle. He's more working on sedimentary environment, marine and freshwater. Also, he has sometimes making incursion into crust habitats, continental or oceanic. And Mark fell into bioturbation during the postdoc in Denmark, so I don't know what place. We will tell us, Mark. And he's sticking to bioturbation. And in the frame of this work that Mark will tell us about how macrofauna affect microbial community structure. Thank you again, Mark, for this talk you're giving to us.
Thank you, Frank. And yeah, it's a great honor to be invited to give a talk as part of this society, which I've only heard about in the past. And we had printed out a poster of the Nereus Park in our lab at ETH Zurich. And we were always in awe of this society, but it's really nice to also then be able to present here. And I'm also a little feeling a little nervous because I've never spoken to an audience consisting of bioturbation experts. So without further ado, I'll today be talking about the role of macrofauna in controlling microbial community structure in marine surface sediments. And my talk has three components. First, I'll talk about, in general, what my interests are, what kind of research I do, and then how bioturbation kind of came into the picture. Then I'll talk about our previous work at ETH Zurich on microbial communities and the bioturbated interval in coastal and marginal sediments, and then about some case studies with the lugworm Aperonicola pacifica in the Pacific Northwest of the US. So my research focuses on the general question, what determines the fate of organic carbon in the biosphere, and I'm looking at mainly aquatic environments, and within those aquatic environments at the transformation of different building blocks of the organic carbon pool, mainly by microorganisms, and the role that microorganisms play in determining the long-term fate of this organic carbon once it gets deposited and potentially buried in the underlying sediments. So most of my work is on coastal and open ocean sediments, including deep subsurface sediments with the ocean drilling program. But in Switzerland, we did a lot of work on lake sediments, also lake bioturbation. And as Frank has already mentioned, I kind of have delved also into other habitats, including the Earth's crust and mantle, subglacial streams, and aquaculture. But my main focus and biggest passion is probably sediments and understanding what happens to organic carbon that gets deposited to the seafloor. And the key question I'm trying to address through my work is what determines whether organic carbon is microbially broken down all the way to the greenhouse gases, CO2 or methane, which can then be released back to the overlying water and atmosphere, and what determines whether that organic carbon stays buried down there and becomes part of this vast carbon sink in marines and other sediments. So in order to tackle this general question, my group focused on studying the microorganisms that are involved in this breakdown of organic carbon. So looking at the different species and of bacteria and archaea and their metabolic potential. We combined that with experiments and analysis of genes that are involved in the breakdown of organic carbon, and perform assays in the laboratory to study the activity of microorganisms and their growth on different organic carbon matrices or substrates. And then we combined that with analysis of the organic carbon pools using organic geochemical techniques to look at what's actually there in the sediments, which components are being buried, which ones are being microbially broken down. And by combining this knowledge on the microorganisms, the organic carbon stocks, and also the processes, we try to understand what the mechanisms are that determine whether organic matter stays buried or is released back as CO2 or methane. And there's a strong quantitative element here looking at both the rates, but also the population sizes of microorganisms and of the different pools of organic compounds. So as a microbiologist, there's been a lot of research in the past decades, especially, but not only, but there have been major breakthroughs since the advent of DNA sequencing tools that have really enabled us to study the vast majority of microorganisms that live in sediments that were previously not, couldn't be studied because they were not culturable. And these studies have shown that there have been thousands of publications that temperature, so the sedimentary temperature, the overall energy supply, how much is there, and also the redox and electron acceptors that are available, such as whether oxygen or sulfate or iron oxides are present, have a strong impact on microbial communities, as well as also the sources of organic carbon, or what's oftentimes referred to as a kind of a black box term, organic carbon quality, which usually refers to either how fresh your organic carbon is or whether it's easily degradable, for instance, of algal origin or terrestrial plant origin. So there's been a lot of work on this, but one major process that is very, very important, as I don't need to tell you, is bioturbation. And this process influences not temperature, but it influences the microbial energy supply, it influences electron acceptor availability and redox conditions, and also the organic matter that is present in sediments. So it has a very strong influence on all of these very important variables that drive microbial community structure. And yet that's been largely ignored by microbiologists in the past decades, or it's just not studied as much as perhaps it could have been. And so the hypothesis that I had that evolved when I was at Aarhus University in Denmark as a postdoc was that bioturbation strongly impacts microbial community structure. And this seems like a no-brainer, but it's interestingly not been studied very much. And the ways in which bioturbation may influence microbial communities and sediment and especially sediment biogeochemistry are pretty known to you, but I'm just going to go over the main ones I'm going to be talking about today. So one process is known as ventilation, which involves the pumping of water in and out of sediments, and with that also the transfer of solutes from overlying seawater, such as oxygen or nitrate, into otherwise anoxic sediments, and also the removal of metabolites that are produced by microorganisms, such as hydrogen sulfide or ammonium, which can become toxic if they otherwise. And so this ventilation can remove these or introduce solutes that have a significant impact on the sediment biogeochemistry. And then there's the sediment reworking, which is the movement of solids, so particles that are in the sediments. And this ventilation can then have a strong impact in that it can cause sediments that would otherwise be anoxic and perhaps sulfidic to become oxic as a result of the strong input of oxygen, or they can change, the redox conditions can change from a reducing environment where we have accumulation of hydrogen sulfide and ammonium to one that is oxidizing, where hydrogen sulfide and ammonia are not present in dissolved form, or almost not, but instead we have iron oxides that would otherwise be removed, reduced largely under anoxic conditions. And it can also cause redox fluctuations. And so these fluctuations between oxic and anoxic conditions, which for many microorganisms are physiologically very challenging, because many microorganisms can be classified as either obligate aerobes or obligate anaerobes. So living in an environment that goes back and forth can be challenging for many bacteria and archaea. Sediment reworking is another very important process, as I don't need to tell you, but this can cause many different things. So one is what's called as biodiffusion, or local mixing, where we have fauna that are moving through the sediments and in the process kind of randomly almost mixing the sediments, causing deeper layers that were maybe under anoxic conditions to suddenly be exposed to oxygen. And then there's also so-called non-local mixing, which is oftentimes related to feeding activity, where, for instance, in this case, it's a certain species of polychaete that feeds on fresh organic matter at the sediment surface and then defecates at a depth of 15 to 20 centimeters below the seafloor, and therefore introduces organic carbon there. And there are also others, so-called upward conveyor feeders that perform the reverse process, that are feeding on deeper sediment layers and then transporting those oftentimes in organic matter depleted form to the sediment surface. I'll be going into those later. So these impacts can be quite dramatic. So on microbial communities, and there's been a lot of work on how microbial metabolism is influenced, and it's been shown that ventilation promotes the aerobic respiration and also indirectly denitrification. And and it can have a strong impact on anoxic or anaerobic processes such as manganese and iron reduction or sulfate reduction rates. And it's been also shown that reworking, so sediment mixing, can either increase organic matter availability or can also decrease it by increasing mineralization rates. But when it comes to microbial community structure, much less is known. So historically, in the early 2000s, there was quite some work on how microbial communities that live in the burrow walls of worms and other macrofauna are affected. And it was shown that growth of aerobes can be promoted in these burrow walls because of the pumping of oxygenated water. And it's also shown that locally growth of nitrogen and sulfur cycling microorganisms can be promoted. So with nitrogen cycling, it's mainly nitrifying organisms, and with sulfur cycling, it's mainly sulfur oxidizing. But not only, also sulfate reducing, so anaerobic organisms can be promoted in their growth. However, there's not much research, much detailed research on how microbial community structure is affected, and most of the work was focused on the actual burrows or sediments directly surrounding the burrows. This then changed, or when I was a postdoc in Denmark, where we kind of by accident stumbled upon this important, what I think is an important question, and that is, to what extent do effects of microbial communities or do effects of bioturbation on microbial communities extend beyond the immediate surroundings of burrow walls? And this brings me to the research that was done in Aarhus and then continued in Switzerland on microbial communities and the bioturbated interval in coastal and marginal sediments. So while I was in Aarhus, I had a doctoral student, Xihan Chen, who's shown here, who is doing research on Aarhus Bay in Denmark. And this is a very well-studied, biogeochemically well-studied coastal bay. And in this study, we were specifically trying to understand what controls the relative abundances of bacterial and archaeal populations in marine sediments. And this has been a question that's been addressed by many studies, but with no consensus in the past decades, where some people have argued that archaea will dominate in in subsurface sediments where energy availability is very low, whereas bacteria dominate in more energy-rich surface sediments. But there's no consensus, and different studies and locations have resulted in different outcomes. But this was kind of how we started this research, and we extracted DNA from sediments and then used quantitative PCR to quantify archaeal populations based on 16S ribosomal RNA genes. And we looked at surface sediments, so the upper 20 centimeters and then down to 10 meters below the seafloor. And what's interesting about Aarhus Bay is that we have very different intervals here. We have surface sediments where we have a lot of macrofauna, and then we have deeper sediments where we have transitions from sulfate reducing to methanogenic sediments. And then in the deepest layers, we oftentimes find a terrestrial layer from a terrestrial swamp of that was present there during the early Holocene when Aarhus Bay was above sea level. Well, what we found was quite interesting in that we saw this very interesting change, marked change in the archaeal copy numbers in the upper 20 centimeters, what looked like an increase in abundance and a strong decrease in bacterial gene copy numbers. And we then calculated the ratios of bacterial to archaeal gene copy numbers and saw that there was an almost 2 order of magnitude decrease in the ratio of bacterial to archaeal 16S genes in the upper 20 centimeters. We're wondering what might be causing this very strong change in the ratios between these different domains. And one idea was that this could be related to the depth of bioturbation. So we contacted scientists at the University of Copenhagen, Torbjørn Andersen, who led to 10 unsupported distributions and was able to show that the depth of sediment mixing went roughly to 18 centimeters, which about roughly corresponded to the depth at which we saw this change in ratio from 100 to 1 almost at the sediment surface to ratios of 2 to 1 or 1 to 1 throughout the remaining sedimentary interval. We also measured pore water chemical gradients, and those also suggested that there was significant introduction of seawater down to roughly 20 centimeters. So this seemed to match this change in the relative abundances of bacteria and archaea. We then performed some not very detailed sequencing of 16S ribosomal RNA gene sequence, and we found that there was also a change in the archaeal and bacterial community structure from this bioturbated surface sediment here at 5 centimeters depth compared to 4 different depths that were the underlying sediments, and these were sulfate reducing sediments, methanogenic sediments, a terrestrial soil layer, and also here a layer where we have the sulfate methane transition zone, where almost all the methane is depleted, that gets produced in deeper layers. And we use 3 different PCR primers, and nonetheless, this is rather robust that we have this strong change from the bioturbated surface sediment to the underlying layers. And what was really interesting is that so-called nitrosopumelalis, which is an order of aerobic nitrifying archaea, was almost exclusively present in the upper surface in the bioturbated layer and not found below. And whereas other groups of archaea that are typically viewed as strictly anaerobic, such as methanomicrobia, and those are the methane cycling archaea and Bathyarchaeota, increased in percentages below. We did the same for bacteria, and they found a similar trend, even more clear, I would argue, that we had a very different bacterial community structure in the surface sediments compared to these other deeper layers, which are actually sedimentologically and geochemically very different from each other, but more similar to each other compared to this bioturbated sediment at the sediment surface. So this resulted in then this hypothesis that the bottom of the bioturbated zone could be an important ecological transition zone where we have a shift from so-called bioturbated or surface sediment microbial communities to subsurface microbial communities. And that led then to the work of Longhui Deng, was a PhD student in Zurich, and he was fortunate to be able to engage in a continual collaboration with Danish scientists and look at the differential impacts of macrofaunal ventilation, and we're working on microbial community structure and marine sediments. And we were at ETH Zurich at the time, and this was funded by the Swiss National Science Foundation. So the cruise that we were able to participate in was in 2014, the geomicrobiology, the Kattegat-Skagerrak seabed expedition with the research vessel Aurora. And this cruise, we visited 4 different locations, 1 in the Lillebælt region and 3 in the upper, around the northern tip of Denmark in the Skagerrak and Kattegat. And we were lucky to have Erik Christensen on board, who is a macrofaunal expert, who then studied the community structure of sediment in fauna at these 4 different locations. And he found that the site that had the greatest water depth, which is in the northeastern part, in the Norway Trench, had very different macrofaunal communities from the sites that were at shallower depths on the in the Kattegat region. And what was also interesting is that our southernmost site in the Lillebælt region had anoxic bottom water, was sulfidic all the way to the sediment surface, and didn't have any macrofauna, making it a very nice non-bioturbated control location. So in order to then study how macrofauna may be influencing the sediment biogeochemical processes, we modeled the non-local exchange coefficients, which give us an idea of the pore water rates of pore water exchanges by macrofauna by ventilation. And we also modeled sediment reworking rates based on lead-210 and also chlorophyll A distributions based on non-local mixing coefficients. And based on these measurements and modeling, we were able to see that there were some clear differences in the depth of bioturbation between the 3 bioturbated locations, ranging from only 8 centimeters at the deep site in the Norway Trench to depths of almost 40 centimeters at the other 2 sites. What we could also see is that based on the ventilation rates, that there were highly ventilated surface sediments in all 3 locations, and then deeper layers where ventilation was minimal, but there was significant reworking based on lead-210 distributions. And so therefore, based on these observations, we subdivided the bioturbation zones into a ventilation-dominated zone in the upper portion of the bioturbated layer and a deeper reworking-dominated layer. Then we want to see how do microbial communities change or compare between these different locations. And what was very striking was that when we compared bacterial communities of bioturbated surface sediments, so in dark blue are the ventilated layers or ventilation-dominated layers, and in light blue, the reworking-dominated layers, the communities of bacteria were very, very similar across all 3 stations that were bioturbated within this uppermost bioturbated layer. And very different, and actually very different from the underlying non-bioturbated layers, and also different from the station that was non-bioturbated at all. There's one small exception, that's the very sediment surface of the non-bioturbated station, which kind of is in between. But other than that, the bacterial community structure is very different at the non-bioturbated deeper layers and non-bioturbated station. For archaea, we saw a similar thing, and that again, different communities were present and actually very similar, again, between the different locations in the ventilation dominant layer and also within the reworking dominant layer. But interestingly, also, we saw that between these two different layers in the bioturbation zone, we saw pretty clear differences between the ventilation dominant layer and reworking dominant layer in archaea. And these differences were less clear, but also there for bacteria. We then used a path analysis to investigate statistically what are the most likely drivers of bacterial and archaeal community structure. And I should say here, we studied the community structure at the zero operational taxonomic unit level. And we found that based on our calculations, reworking through its impact on the distribution of organic matter was the process or variable that explained most of bacterial community variation, whereas ventilation, so the pumping of oxygenated seawater into the sediments, explained most of the archaeal community variation. And this is quite interesting because it suggests that bacteria and archaea are influenced or controlled by different bioturbation-related processes in these sites. In the underlying subsurface sediments, we found that, or our correlations indicate that bacteria, similar to surface bioturbated layers, are strongly controlled by organic carbon, isotopic compositions, and total organic carbon, whereas for archaea, that's also the case, but most of the variation in archaeal community structure and subsurface sediments couldn't be explained with the measured variables. So this brings me to a brief synopsis of this first two studies that I presented. And the first point is that based on our analysis, bioturbation drives microbial community structure from the domain to the operational taxonomic unit level in surface sediments, whereby ventilation, which mainly probably by controlling oxygen and or redox conditions, is the main driver of archaeal community structure, whereas reworking, and there we found the strongest correlations with isotopes and also the content of fresh algal organic matter based on chlorophyll A measurements. Reworking seemed to mainly drive bacterial community structure through its influence on the distribution of fresh organic matter in surface sediments. It was interesting to see that so-called subsurface taxa that were initially discovered in the deep sea and have been since studied a lot during ocean drilling expeditions and are found in sediments that are tens to hundreds of meters below the seafloor, dominate everywhere in these sediments as soon as we're outside of the zone of bioturbation, and even in surface sediments of sites where we don't have bioturbation. So we shouldn't call them subsurface taxa, as has been done widely in the subsurface microbiological community, but perhaps we should refer to them as non-bioturbated sediment taxa. And based on our analysis here, it seems that the main driver of the community structure of these subsurface taxa or non-bioturbated sediment taxa is related to the sources or chemical compositions of the organic carbon. So with that study on these different locations in Danish waters, I'm going to shift now to a study or several studies that were done on a single species of worm, the lugworm Aporenicula pacifica in the Pacific Northwest of the US. And this was work that was done at the Friday Harbor Marine Laboratories at the University of Washington. And while all the previous work was done in subtidal systems, we decided we want to do some intertidal work that allowed us to actually easily visit the sites of bioturbation and make it easier for us to perform experiments and study the impact of bioturbating fauna on microbial community structure in sediments. So the location studied is up here in the Pacific Northwest, very close to Canada, state of Washington, and we were looking at a particular location on San Juan Island that's known as False Bay, which has been a very well-studied location in terms of bioturbation and bioturbating macrofauna, which is why we decided to go there all the way from Switzerland. And we looked at mainly at this False Bay Marine Preserve, which I just showed you, and performed both field research on Aporenicula pacifica and also then some laboratory research, in which we performed experiments both with Aponicula pacifica and Nereis vexillosa, so a different, an actual clamworm, so a worm species with very different behavior. Because I don't have enough time to go over both, I'm going to focus on the field study, but if you have any questions, I'm happy to show some slides from the laboratory study also. So this location on San Juan Island, and in particular False Bay, has been a site of major discoveries in the area of bioturbation research. It's where Jørgen Hylleberg, a Danish scientist from Aarhus University back in the 1970s, proposed the concept of microbial gardening in this paper. And it was based on Aponicula pacifica, which is this lugworm that lives in the intertidal area of False Bay, and based on chemical measurements of N-organic nitrogen compound distributions, it was proposed that Aponicula pacifica enriches the sediment of its feeding pocket by stimulating the growth of microorganisms, which it then feeds on subsequently. Another study that was also performed not far away, just slightly further north on San Juan Island, resulted in the proposal of algal gardening by nereid worms. And here was observed that different species of nereids attach pieces of drift algae, so different members of the genus Ulva and Enteromorpha, which I guess nowadays is also considered Ulva, were attaching this drift algae to the tube surfaces and then causing the algae to attach and grow there. And then they were feeding on the growing portions of these macroalgae. But today I'll be only focusing on Aponicula pacifica and kind of pursuing further this idea that microbial gardening may be performed by Aponicula pacifica in its feeding burrow. And we did this based on intertidal research in a certain location of False Bay where Aponicula pacifica almost occurs in a monoculture. There is almost no other macrofauna present in this location. And there were fortunately also immediately adjacent areas where no macrofauna were present that were geochemically very similar. Those were our control areas. So Aponicula pacifica is a typical lugworm in that it produces this J-shaped burrow, which has a feeding pocket down here, and it's slightly above that where a microbial gardening is supposed to be happening according to the previous research. And above this feeding pocket, there's usually a feeding funnel, which is a slight depression at the sediments, which you can see here in a few locations, here especially well, and that's where the sediment is collapsing as a result of the Aponicula feeding and ingesting sediments here, which it then defecates out at the sediment surface as fecal casts, which are everywhere here on this photo. Aponicula pacifica not only performs this kind of upward conveyor feeding, but also has a strong impact on the pore water chemistry because it very actively pumps water from the overlying seawater into its burrow and into the overlying sediments. And it can also even reverse the flow of the water out of its burrow again at times. So we were interested in, first of all, just characterizing the sediments geochemically, sediments with Aponicula pacifica with very high densities. I forgot what the numbers were, but I think it was 35 a specimen per square meter or in that range, and to an adjacent area where these worms were absent. And the first thing we did was look at the grain size distributions, and those are shown here. And you can see that sediments were dominated by silt and sand. And while surface sediments were very similar in terms of the relative contributions of silt and sand, we could see that there was a strong depletion of the silt fraction in sediments where Aponicula pacifica is feeding, so where the feeding pocket is of this worm. And this is not a new finding. It's been shown previously that Aponicula pacifica preferentially feeds on fine organic particles, but that also includes silt and clay fractions, and then defecates those out at the sediment surface, while at the same time selectively not eating the sand and gravel portion. And that results in so-called graded bedding and enrichment of this more coarse sedimentary material right at the living depth of Aponicula pacifica. What this then led us to propose was that there are different vertical zones within sediments that are inhabited by Aponicula pacifica and adjacent areas where there's essentially no macrofauna. So we distinguish between a physically and biologically controlled surface layer, a biologically controlled deeper layer in upper Ornithola pacifica sediments, and in deeper undisturbed sediments where bioturbation activities were no longer detectable. For the adjacent area, we only came up with these two definitions. There's the undisturbed sediments where there's no influence of physical mixing, but then there's also a significant upper sediment layer of 10 to 12 centimeters thickness, which is strongly physically controlled by movements of the tides that can cause significant mixing of the sediments and also physically induced pore water ventilation. So we then want to see what is the impact of Upper Ornithola pacifica on microbial communities, but to really understand how these worms are influencing microbial communities, we need to perform an in-depth characterization of the geochemical impacts. So the first thing we did was look at the changes in pore water geochemical gradients, and we looked at a wide range of analytes and found that based on just concentration profiles here, bioturbated and non-bioturbated sediments, which are shown below, are not that different. We found significant lower ammonium concentrations, especially in the deeper layer of the bioturbated sediments, so this biologically controlled layer. We found slightly lower iron concentrations, and manganese concentrations were considerably higher in this deeper layer, but we're talking about very low manganese concentrations in the low micromolar range. For sulfate and hydrogen sulfide, the impact was a little stronger. So for sulfate concentrations, we saw overall slightly higher, a few hundred micromolar higher concentrations throughout the sediments, and then in the non-bioturbated sediments. But for hydrogen sulfide, the impact was pretty clear in that we measured much lower hydrogen sulfide concentrations in the bioturbated sediments, which can be explained with both the flushing of the sediments by the worms and the introduction of oxygenated seawater, which then chemically oxidizes hydrogen sulfide, or can also stimulate the biological oxidation of hydrogen sulfide. For dissolved inorganic carbon, we saw this interesting slight increase in dissolved inorganic carbon concentrations, but actually the profiles were not that different. We see a slight decrease in the non-bioturbated sediments. But nonetheless, based on these data and some experiments that we performed in which we artificially removed Upper Ornithola pacifica, we came up with this categorization of the sediment into an upper sediment interval where there's significant physical mixing, and that's based on coefficient of bioirrigation measurements on DIC, so dissolved inorganic carbon, and then a deeper layer where there's significant pumping of seawater into the sediments that's occurring by the worm. In addition to these pore water gradients, we want to see what is the impact of deposit feeding, since we know that Upper Ornithola pacifica is a very active deposit feeder, and we want to see how does this deposit feeding change solid phase geochemical gradients. And here we saw a much stronger impact. So we see that total organic carbon contents are much lower in the upper sediments and also in the biologically controlled layer, except for the deepest part of this biologically controlled layer, than in the non-bioturbated sediments. Interestingly, also, we see very different profiles of carbon to nitrogen ratios. So we see this very strong increase in carbon to nitrogen ratios to values of 30 at the right below the feeding pocket of the worms. And it was interesting to note that also the isotopic compositions of the organic carbon were very low there. There's one explanation for this, and that this is also matched by physical observation. We found a lot of woody debris right below the feeding pocket of these worms. And so what this suggests is that Upper Ornithola pacifica is feeding very heavily on the labile microalgal organic carbon, but is not able to eat or avoids eating the larger macro debris of vascular plants. And as a result of that, we have this strong enrichment of this debris in these deeper layers where they become buried right below the feeding depth of the worm. We wanted to also look at the more labile portions of the organic carbon, and their chlorophyll A and phenolpigments are oftentimes used as proxies for organic carbon at different degradation states, with chlorophyll A being kind of the fresh organic carbon and phenolphyte in the slightly more degraded portion. And while chlorophyll A profiles were similar between the two different settings, the bioturbated and non-bioturbated sediments, for phenolpigments we saw a big difference. So we see that phenolpigments were very, very low in the sediments with lugworms, compared to the sediments where there's no bioturbation present. And one explanation here is that these lugworms are constantly feeding and ingesting sediment, and in the process, not only removing chlorophyll A, which is a renewable resource, which regrows very quickly, but also removing the detritus that's present, which accumulates significantly in non-bioturbated sediments, but gets removed by digestion and expulsion at the sediment surface in these lugworm-dominated sediments. This is also reflected when we calculate the ratios of chlorophyll A to few pigments, where you can see an even bigger difference between the bioturbated and non-bioturbated sediments. And then we, as microbiologists who like to work on DNA, we also quantified gene abundances of macrophyta, which in this case were dominated by microphytobenthic diatoms, and compared those to sediments where lugworms were not present. And here the striking thing is that surface sediments are similar, but subsurface sediments have several orders of magnitude lower diatom gene copy numbers in the bioturbated sediments than the non-bioturbated sediments. And again, we think this is because these lugworms are constantly driving down the sediment and removing their preferred food choice, which are microphytobenthos from these sediments. Interestingly, also vascular plant gene copy numbers were strongly depleted in sediments with Aporenicula pacifica compared to the control treatments. And most likely that's because we have the removal of labile organic matter from seagrass beds, which are nearby in these sediments, but we didn't pursue this follow-up further, so I cannot really say for sure which portions of the plant DNA is being removed here. We then used data again from our parallel experiments with defaunated plots and refaunated plots to look at sediment mixing rates, and we were able to show that there's a significant physical mixing rate that occurs in these sediments of false bay that's caused by the intertidal movements that can stir up the sediments and move the sediments around. But we also could see, based on chlorophyll A distributions, that there's significant subsurface deposit feeding and therefore also subsurface reworking occurring in the sediments with Aporenicula pacifica. Now, looking at how this impacts microbial community structure, we first looked at the abundances of bacteria and archaea, and additionally, also we looked at the abundance of eukarya. And here now I'm showing the bioturbated sediments and non-bioturbated sediments together. And what you can see is that both bacterial and archaeal gene copy numbers were significantly lower in bioturbated sediments compared to the non-bioturbated sediments, with the biggest impact seen in the areas of the depth layer where Aporenicula pacifica are feeding, where we saw up to 2 orders of magnitude lower bacterial and archaeal gene copy numbers than in control sites. And this is consistent with the selective removal of fresh particles, organic-rich particles by the worms, and in the process, the breakdown and consumption and digestion of bacterial and archaeal populations. Interestingly, for eukarya, we saw a bit of a different trend, that we actually saw significantly higher eukaryotic gene copy numbers in the uppermost layer of bioturbated sediments. And one possible explanation is that Aporenicula pacifica is pumping oxygenated water into these uppermost sediments, and therefore enabling or stimulating growth of certain types of eukaryotes that are perhaps mobile and therefore can avoid being drawn down and consumed in these deeper layers. Nonetheless, we see this strong depletion of also eukaryotic DNA in these deeper layers, which is probably mainly because of the removal of eukaryotic microalgae. Then looking at the impacts on certain metabolic groups of bacteria and archaea, we mainly focused on the sulfur and nitrogen cycles, looking at the reductive sulfur cycle, sulfate reduction, which has a key gene, DSRB, and sulfide oxidation, which is a key gene, SOXB, and then the reductive and oxidative nitrogen cycle, where we have NARG and MOA, which are functional genes that are involved in denitrification and nitrification. What we found is that anoxic or anaerobic, the process genes involved in sulfate reduction were strongly depleted in sediments with Aporenicula pacifica compared to control sediments. And this was also seen for genes for denitrification and for sulfide oxidation genes. But interestingly, for MOA, so nitrification, there was an actual higher abundance of nitrification in surface sediments. And we explain this with perhaps the fact that, again, oxygenated seawater is being drawn into this surface sediment and may stimulate the growth of ammonia oxidizing microorganisms in this uppermost layer. We then looked at the microbial community structure, looking at both richness and using a principal component analysis. And what we found is that richness of bacteria and archaea was much lower in this biologically controlled feeding layer of Aperonicula pacifica, both in bacteria and archaea, when compared to non-bioturbated sediments, which are shown in black here. And whereas the richness was almost not influenced at all in the surface sedimentary layer where the worms are not feeding. For eukarya, we see a much lesser impact. We see slightly higher richness in the, higher richness in the sediments with Aperonicula pacifica and the surface sediments than in the non-bioturbated sediments, but no difference in the deeper layers. Then looking at how the community structure compares, and again, we're looking at 0 operational taxonomic units here. We see no impact on bacterial or archaeal or eukaryotic community structure in the surface sediments between bioturbated and non-bioturbated sediments, but a strong influence in this deeper layer where we have feeding by Aperonicula pacifica as a very important process, which results in red shown here, a very different bacterial and archaeal communities in these deepest layers. For eukarya, the influence is much smaller. We then want to see how does the Aperonicula pacifica impact possibly interactions between different groups of bacteria, archaea, and eukarya. And one way that this is frequently done nowadays is by performing correlation analysis between different taxa. So we did this for bioturbated and non-bioturbated sediments, and we're showing these here at the class level. And interestingly, we could see is that there's a strong influence on the community networks, so the correlations between bacterial taxa by the bioturbating lugworms compared to non-bioturbated sediments, with much stronger correlations between bacterial taxa in the non-bioturbated sediments. And one thing I should also mention here, for the non-bioturbated sediments, we have 2 different clusters, C1 and C2. And C1 are bacterial communities that live mainly in surface sediments, and C2 are ones that are mainly present in subsurface sediments. So there seems to be a clear structuring between surface sedimentary and subsurface sedimentary bacterial communities within these non-bioturbated sediments. One possible explanation is that the surface sediments here are undergoing strong physical mixing, and that selects, even though there's no bioturbation, for quite different bacterial communities in the upper 10 centimeters compared to the deeper layers where there's no impact of this physical mixing. For bacteria in the bioturbated layers, we see different pattern, and we actually see even a group of bacteria that are only correlated in bioturbated sediments. And we don't really know what drives these differences in correlation. So this is an interesting question for future research. But one interesting observation was that B3 and B4 are bacteria that can be divided into algae degraders and terrestrial plant degrading bacteria. And those showed a clear separation in the bioturbated sediment layers between the feeding pocket, where we have input of fresh algae from above, and the sediments below the feeding pocket, where we have accumulation of terrestrial debris. So there's something interesting there, but we have to do more work to understand what's actually happening. Remarkably, for archaea, there was a much smaller influence on these networks of archaea by Aperonicula pacifica, whereas for eukarya, on the other hand, we saw a really dramatic impact. So we see very few consistent correlations between eukaryotic taxa in the non-bioturbated sediments, but we see very strong correlations between eukaryotic taxa in the bioturbated sediments. And here it's again mainly in surface sediments where we see a strong impact. And one explanation is that in the bioturbated sediment layers, we have a strong selection for algae degrading microorganisms and smaller eukaryotes that are potentially feeding on the algae and algal detritus. However, this is, I would say, still speculative. And we think that the reason why this could be important is we find higher abundances of microeukaryotes in the surface sediments of bioturbated sediments, and perhaps their presence there is stimulated or enabled by the pumping of oxygenated water by Aparonicula pacifica. So with that, I'll provide you a brief summary of this second component of my talk. So Aparonicula pacifica, based on our research in False Bay, is the main driver of organic carbon cycling in subsurface intertidal sediments of False Bay. So the upper 10 centimeters, the impact is much smaller, and that's because we have strong physical forcing there as a result of this part of the intertidal zone becoming exposed to UV radiation, strong heating, and also physical mixing by the tides. And this is all happening during the summer months. But then below those the upper 10 centimeters, we have a very strong impact on the organic carbon cycle by Aparonicula pacifica. And though we see that ventilation significantly changes pore water chemical gradients, it seems that the deposit feeding by this worm in its feeding pocket and living depth has a much greater impact on the carbon cycle in these sediments. We also observe or see clear evidence that Aparonicula pacifica removes microalgal detritus and strongly lowers microbial abundance in these subsurface sediments, and in turn, by selectively avoiding woody debris, increases the burial of woody debris in these sediments. We also see that selective feeding on microalgal organic matter alters bacterial communities, and that's mainly by impacting bacterial communities that are involved in microalgae or algal degradation, and also that bacterial and eukaryotic community structure is strongly influenced, whereas archaeal community structure is much less influenced. Another remarkable finding, and which is something we also observed in the continental shelf regions off the coast of Denmark, was that bacterial communities that we found here, when we performed further analysis of their genomes based on genomes of closest relatives, it appears that bacterial communities in the bioturbated sediments are dominated by microaerophilic and also facultatively aerobic taxa. So taxa that can live or require low concentrations of oxygen or can even switch between anoxic and oxic conditions and are adapted to this kind of fluctuating redox conditions or oxygen concentrations. And those seem to be dominant in the bioturbated sediment layers, whereas for archaea, we don't see that, any evidence of that. And that brings me to the final conclusions. Based on our work, we think that macrofaunal bioturbation is perhaps the most important driver of microbial community structure in marine surface sediment. The impact of bioturbation on microbial communities varies with macrofaunal species, so the impacts of the lugworms are not necessarily what we saw on continental shelf environments. Nonetheless, there's everywhere we see clear evidence for a strong impact of bioturbation on microbial communities. We also see that bacterial communities seem to be mainly correlating with reworking related impacts, which strongly influence the distributions of algal organic matter, whereas archaeal communities are more strongly affected by ventilation. And one working hypothesis is that archaeal communities, unlike bacteria, are not as adept at coping with redox fluctuations or fluctuating oxygen concentrations. So maybe they can be divided into strictly anaerobic taxa and aerobic taxa, and therefore bioturbation has a very strong negative impact on archaeal communities, and also this could explain why ventilation and not reworking is the most important driver of archaeal community structure. With that, that was my talk, and I'm happy to take any questions from you.
Stefan? For the first, I saw the head, Stefan. You can go.
Okay, can you hear me? Okay, well, thanks very much for a very a talk encompassing many aspects. It's very great to listen to it and to watch it. You were talking about this layer, 20 centimeters, well, the feeding layer of Aparonicula, where you had 2 orders of magnitude lower numbers in most bacteria, I guess. do you know somebody who has any idea on what the rates of activities in those bacteria are? Because numbers is one thing, and maybe they're stimulated. I don't know.
Yes, actually, and this is something I didn't present to you. We performed laboratory incubations at Friday Harbor Laboratories with Aperonicola pacifica and also with Nereis vexillosa. And we found that net rates of aerobic respiration and denitrification were higher in sediments with Aperonicola pacifica, whereas sulfate reduction rates were lower. And overall, so we think there is a strong, very dramatic stimulation of bacterial activity by Aperonicola pacifica, and that compensates, even though we have much lower populations, the rates of aerobic respiration and denitrification are probably higher overall in sediments with Aperonicola pacifica.
Yeah. Okay, thanks.
Any question?
And I should mention this is a paper that's submitted and will come out hopefully in the near future. So if you, maybe in the next few months or so, it can be seen.
I have a question. So Aperonicola is pretty unique in the effect that it has on sediments in terms of different types of macrofauna. And I was wondering what you think the difference might be in a, say, a macrofaunal taxa. You alluded to this in your conclusions, but something that burrowed a bit differently, like, for example, nereids that might create these sort of networks of burrows or tunnels extending out to the sediment, and sometimes they line it with a mucus that might have effects on the bacterial community as well.
Yeah, so we did perform these experiments in the laboratory with Nereis, and we found that the mucus community is completely different from that in surrounding sediments.
And also from that in that you would see in the lugworm as well? Is it in the same type of sediment or?
So the lugworm didn't line its burrows with mucus, so we did not look at that.
Okay, all right. So the microbial communities of the lugworm sort of exterior, the lining, yes. Not the lining, but the,
we performed experiments for, maybe I should share this if you want to see the results from this study. Let's see. So can you see it?
Mm-hmm.
So we looked at Nereis vexillosa, NV, and Aperonicola pacifica, and compared feeding funnel sediments. So we took a core right basically through the feeding funnel from the top and then adjacent areas. And what we found is that bacterial community structure was completely different when we looked at the mucus of Nereids, Nereis vexillosa, which is shown here, these two dots, and was also very different in the fecal piles of Aperonicola pacifica. When we looked at, but we basically removed those outliers and then looked at, well, other differences in the sediments with Nereis, which are shown in blue, compared to sediments with Aperonicola pacifica. And I should say
it's the first slide of your talk, the title slide.
Sorry. Oh, okay, now let's see. Here we are. Can you see now?
Yes, yeah.
So what you can see in black are the control incubations without fauna, and you can see similar microbacterial community fingerprints in blue in the Nereis sediments. And I should say here, the solid blue colors are the burrow sediments, and the non-solid, so the empty blue are sediments that are further away from the Nereis tunnels. Or burrows, versus for Aperonicola pacifica, we see different. It's a bit messy, but we see distinct bacterial community fingerprints both in the feeding funnel of the worms, but also further away from the feeding funnel. So we see clear impact on bacterial community structure essentially throughout these aquaria that we used in Apernicola pacifica treatments, but for nerids, it was very much restricted, and there's only very minor. It was mainly found in the mucus that was present. And we explained that we modeled the bioirrigation rates, and we found that bioirrigation of nerids was mainly limited to, well, the actual tube itself, and within a few millimeters or at most centimeters of the U-shaped tunnel. Whereas for Apernicola pacifica, we saw a complete change in the pore water geochemistry, which this was done on narrow aquaria, but just to give you an example here, here's a control aquarium where we had 6, I think this was probably ammonium concentrations. Yes, very high ammonium concentrations. In the nereid treatment, we have strong removal of ammonium, but still accumulation to millimolar concentrations in sediments that are further away from the tunnel. And for Apernicola pacifica, we have a complete change in the geochemical environment with ammonia concentrations only a few hundred micromolars high. And this was also seen for hydrogen sulfide, same picture, with hydrogen sulfide being essentially non-detectable throughout Apernicola aquaria compared to it's being locally present in the Nereis vexillosa treatments to several hundred micromolar. So this is a long answer, but we see much stronger impact of Apernicola pacifica.
Yeah, and it's not creating those sort of micro habitats that other animals that wouldn't be flushing as completely are doing. That's really interesting. Yep. Thank you.
Anyone? Question? Okay, so I have one. Mark, it's about the when you show us that depending on this process. Okay, sorry, I have to start the camera. It's more polite. I know. I can't. Sorry. That bioturbation was explaining more the reworking was more explaining the change in bacteria, and the ventilation more the change in archaea. And I was just wondering, I like classification. If we knew different species and having a ratio between the way they rework versus the way they ventilate as an intensity, could we extrapolate to say that this species will more affect archaea than bacteria?
I think
and then if we go further, if we mix species, say that, okay, this community we create will more affect bacteria than archaea? I
think that's the implication. And it would be very interesting to think of where would be good locations where we have the right, I mean, I guess we could test it experimentally in the laboratory, or go to natural locations where we have very big differences in terms of ventilation versus reworking activities. We tried to do this actually in Swiss lakes by comparing sediments where we had mainly oligochaetes, so tubificids present, which are more reworking, and lake sediments where we had chironomid larvae, which are mainly ventilating. Unfortunately, the impact was very, very small. And we also did experiments in the laboratory that are published. There was only a minimal impact of these chironomids and oligochaetes on microbial community structure in the 3-month incubations that we performed, and also in the field. So I wonder why that is. If it could be related to the size of the macrofauna, that in the marine environment we have oftentimes much bigger. We have very large macrofauna that are have a much stronger geochemical impact by completely flushing the sediments, and something we don't find often as much in lake sedimentary environments, at least not the ones that we studied. So if you have any ideas for places to go to in the marine environment that would kind of provide this kind of difference between reworking versus ventilation dominated, would be, I think, very interesting to look at.
Any question? Anyone else? Okay. So thank you, Mark, again. Thank you all for attending. And yes, thank you, Mark, again for proposing to give us this talk. Next talk is planned for 12th September. I guess. Yes, something. Yes, 12th September, and we'll have a talk from Nice Volkeborn. That is actually at Stony Brook. And for those who receive my email from announcement or whatever, just the information that I'm trying to have a real mailing list that each of us could use it to send to the community message. So far, it's just me sending message because I have trouble with the list. The thing is to have a email list that you can sing in and sing out, and you can use it to send by yourself message to the community. Okay, guys, thanks again. Thanks to Agrient for setting up the seminar. Perfect as always. And yes, I want to say just say Kelly, Mark Kelly couldn't make it, so she apologized. She couldn't make it. So okay.